Louis Pasteur, a name that echoes through time. He changed our world, in so many wonderful ways. This is his story, the story of a remarkable mind. It begins in a quiet corner of France, in a small, unassuming town. The year was 1822, December the 27th. A world very different from ours, full of unanswered questions. Science was still in its infancy, and many mysteries remained unsolved. But a beacon of light was about to appear from these simple origins, a mind that would illuminate the darkest corners of disease and offer hope to millions. He was born in the town of Dole, nestled in the Jura region of eastern France. His father was Jean-Joseph Pasteur, a tanner by profession. This was a traditional craft, turning animal hides into leather. Jean-Joseph had also served his country as a sergeant major in Napoleon's armies. From his father, young Louis learned about duty, about perseverance, and about patriotism. The Pasteur family lived a modest life. They were not people of great wealth, but they understood the value of hard work and the importance of character. These early lessons would shape Louis throughout his extraordinary life. Life for the Pasteur family soon moved from Dole to nearby Arbois. This was the town Louis would always consider his home. A place of vineyards and rolling hills, a picturesque setting. Here, young Louis grew. A serious child, they said, thoughtful and observant. He played like other children, but his mind was already curious. He watched the world around him, the artisans at their work, the farmers in their fields. Little did anyone know then the incredible journey that lay ahead for this boy. The world of science was waiting for his unique and brilliant contributions. The France of Pasteur's early childhood was recovering from years of revolution and war. Society was changing, industry was beginning to grow. But when it came to health and sickness, understanding was very limited indeed. Diseases would sweep through communities, and people often did not know why. Superstition and old theories still held sway for many common ailments. The idea of tiny, invisible creatures causing illness seemed like fantasy to most. This was the world Louis Pasteur was born into, a world ripe for discovery, and he would be the one to unlock some of its most vital secrets. When Louis first went to school, he was not an immediate star. He was a diligent student, but not exceptional, his teachers thought at first. Science, in particular, did not capture his imagination initially. He found his lessons adequate, but no special passion burned within him, not yet. Other subjects seemed to hold more appeal for this quiet, observant boy. He was steady, he was serious, but the spark of genius was still hidden. It needed time and the right moment to truly ignite and shine brightly. The path to becoming a great scientist is often not a straight one. Interestingly, young Louis showed a real talent for something quite different from science. He was a gifted artist, especially with pastels. He drew portraits of his family and his friends. His eye for detail was remarkable. He could capture a likeness with surprising skill. Look at these early drawings. You can see the concentration, the careful hand. This artistic ability revealed something important: his keen power of observation, a quality that would later serve him well in his scientific endeavors. He saw the world with clarity, whether through a pencil or a microscope. His portraits were quite accomplished for a boy of 13, then 15. His mother, his father, his sisters, their faces preserved by his youthful hand. Some people in Arbois even commissioned him to draw their portraits too. Imagine if he had chosen art. He might have become a painter of some note. The world would have known a different Louis Pasteur, perhaps a celebrated artist. But another calling was stirring within him, a pull towards deeper questions. The world of lines and colors would eventually give way to the world of molecules. His artistic eye, however, would remain a faithful servant to his scientific mind. As he grew older, his approach to studies began to change. A new seriousness took hold, a greater determination to learn. Perhaps it was the influence of his headmaster or a growing awareness of his own potential. He started to apply himself more rigorously to his lessons at the Collège d'Arbois. He began to dream of more, of higher education, of making his mark. The world of knowledge was vast, and he felt an increasing desire to explore it. The quiet boy was finding his ambition. The path of science slowly started to beckon. His journey of discovery was truly beginning to take shape. The desire for deeper knowledge grew strong. Louis aimed high for his education. He attended the Collège Royal in Besançon, a larger town, a bigger stage. Here he worked with dedication. He knew his family had made sacrifices for him, and he was driven by a need to succeed, to make them proud. He studied hard, focusing on his subjects with intense concentration. This period was crucial. It built the foundation for his future studies. He was no longer just an average student. He was becoming a scholar. His mind was sharpening, his curiosity was deepening each day. Then came the great challenge, Paris, the heart of France, the city of learning. In 1838, he first ventured to Paris to attend a preparatory school. But the vast impersonal city overwhelmed him. He felt terribly homesick. The young man from the countryside missed his family, missed Arbois. His health even suffered, so strong was his longing for home. He returned to Arbois for a time. It seemed his Parisian dream might end. But Louis Pasteur was not one to give up easily. His resolve was strong. He would return to Paris, better prepared and more determined than ever. His perseverance paid off. He aimed for the prestigious École Normale Supérieure. This was one of France's finest institutions for training teachers and scientists. Entry was incredibly competitive. He had to study relentlessly. He earned his Bachelor of Science degree, and then he focused on the entrance exams. In 1843, he was admitted. It was a momentous achievement, a testament to his hard work. At the École Normale, he found his true calling. Surrounded by brilliant minds, he immersed himself in physics and especially in chemistry. The world of atoms and molecules fascinated him completely. Within the halls of the École Normale, he encountered influential teachers, men like Jean-Baptiste Dumas, a renowned chemist of his time, and Auguste Laurent, another brilliant mind in chemistry. These mentors recognized Pasteur's potential. They encouraged his burgeoning talent, they guided his studies, they challenged his thinking, and they inspired him. He learned the importance of rigorous experimentation, of careful observation. The very skills he had once shown in his art were now being honed for science. The young student was transforming into a scientist on the cusp of greatness. At the École Normale Supérieure, Pasteur's passion for chemistry and crystals blossomed. He investigated tartaric acid from wine sediments which rotated polarized light. Under the microscope, he discovered two types of mirror-image crystals. Separating them, he found they rotated light in opposite directions, solving the mystery. This groundbreaking discovery revealed the connection between molecular shape and properties. Louis Pasteur's groundbreaking work on crystals, it really opened new doors. It showed the world his brilliant mind, his patience, and his sharp observation. But this success, it didn't make him rest. In fact, it spurred him onwards. He was appointed Professor of Chemistry and Dean of Sciences in the city of Lille in the year 1854. Lille was a vibrant hub of industry and production. Beetroot sugar and alcohol distillation were vital local businesses. This new environment would soon present new challenges and, honestly, new opportunities for discovery. Pasteur believed deeply that science should serve humanity. It shouldn't be confined to laboratories or dusty textbooks. He really wanted to solve real-world problems, to help ordinary people. In Lille, he encouraged his students to look at the local industries. Where will you find better subjects for study, he would ask them, than the germination of seeds, the fermentation of grape juice? His own attention was turning, from the symmetry of inanimate crystals to the mysterious processes of life and the changes they brought about. The world of fermentation was calling, a world full of questions. For centuries, people had made wine and beer and bread. They knew that grape juice turned to wine, that barley and water became beer, but how did this transformation happen? What was its true nature? Most scientists believed it was a purely chemical process, unrelated to life. Others thought tiny organisms just appeared spontaneously from non-living matter. These were old ideas, deeply entrenched. But Pasteur was not afraid to question. He approached these puzzles with his characteristic thoroughness and, an open mind. His earlier work on crystals had shown him the subtle power of asymmetry. He had found that living things often produce these asymmetric molecules. Could this be a clue to understanding processes like fermentation? He began to wonder if living organisms might play a crucial role, a role that science had previously overlooked or misunderstood. The industries of Lille, particularly the alcohol producers, were about to provide him with the perfect opportunity to test these emerging ideas and to embark on a new scientific adventure. The producers of alcohol in Lille, they faced a serious problem. Their businesses depended on fermentation, turning sugar from beetroots into alcohol. But often things went wrong, terribly wrong. Instead of good, strong alcohol, their vats would yield a sour liquid or a product that was weak, unpleasant, and, well, unsellable. This meant huge financial losses. It threatened their livelihoods. They were desperate for answers, for a way to control this fickle process. Why did some batches succeed while others failed so miserably? The prevailing theories offered little help to these troubled distillers. Many chemists believed fermentation was a kind of decomposition, a breakdown of sugar caused by instability in the molecules themselves. They thought yeast, often found in fermenting liquids, was just a byproduct, a complex chemical substance perhaps, but certainly not alive, or if it was alive, it was spontaneously generated by the fermenting brew. These explanations gave no practical guidance on how to prevent spoilage. The problem remained a costly and frustrating mystery for the industry. One local distiller, a man named Monsieur Bigot, had heard of Pasteur. He knew of the young dean's reputation, his keen scientific mind. His son was one of Pasteur's students at the Faculty of Sciences. Monsieur Bigot approached Pasteur. He explained the difficulties they faced, the diseases of their beet alcohol, the unpredictable nature of the fermentations. Could the esteemed professor shed any light on this vexing issue? This was exactly the kind of challenge Pasteur sought, a practical problem, a chance to use science to make a tangible difference. Pasteur listened intently to the concerns of Monsieur Bigot. He understood the urgency, the need for a reliable solution. He promised to investigate, to bring his scientific methods to bear. He would visit the distillery, examine the processes, and collect samples of both the successful and the failed batches. This was not just about helping one distiller or one industry. Pasteur sensed a deeper puzzle, one that could unlock fundamental truths about the nature of fermentation, and perhaps about the nature of life itself. Louis Pasteur began his investigation with his most trusted tool, the microscope. It was his window into a world invisible to the naked eye. He had used it before to study the tiny facets of crystals. Now he would use it to search for clues in the fermenting liquids. He collected samples from Monsieur Bigot's distillery, samples of the beet juice, samples of the fermenting mixture, samples of the good alcohol, and samples of the spoiled acidic batches. He was meticulous, careful, leaving nothing to chance. He started with milk, another familiar product that underwent fermentation. Everyone knew milk could turn sour. It was a common experience. Pasteur examined fresh milk under his microscope. He saw tiny globules of fat. Then he examined milk that had soured. He saw something new. Alongside the fat globules, there were tiny rod-shaped particles or little dots. These were much smaller than yeast cells. He observed them multiplying as the milk became more acidic. These tiny organisms seemed to flourish. Was there a connection? Could these minute beings be causing the souring? This observation was intriguing. It hinted at a biological cause, not just a chemical change, but a change driven by living things. He then turned his attention to the beet juice fermentations. In the vats that produced good alcohol, he saw something consistent. Under the microscope, he observed round, budding globules, yeast cells. These yeast cells were plentiful, and they looked healthy, active. This was expected. Yeast was always present in alcoholic fermentation. But what about the spoiled batches? What would he find there? When Pasteur examined the vats that had produced sour alcohol or other unwanted products, he saw a different picture, a very different microscopic scene. Yes, there were yeast cells, but often they looked unhealthy, less numerous, and crucially, he saw other microorganisms, just like in the sour milk. There were the tiny, rod-shaped creatures, similar to those he'd seen before, and sometimes other forms, different from the yeast, different from the rods. These other microbes were abundant in the very batches that had gone bad. A pattern was emerging, a vital clue to solving the mystery of spoilage. So the presence of yeast in alcoholic fermentation, that was not exactly a new discovery. Scientists like Cagnard, Delatour and Schwann had already suggested that yeast was alive. They believed it reproduced by budding and was somehow linked to making alcohol, but honestly their ideas just weren't widely accepted by the leading chemists of the day. Men like Liebig, Berzelius and Wohler, real giants of chemical science, they dismissed the notion that tiny living cells could cause such a profound change. They argued that fermentation was purely chemical, just a breakdown of sugar. Yeast, they said, was just a complex, unstable organic substance, maybe a catalyst at best. But Pasteur's careful observations led him to a different conclusion. He saw that yeast was always present when sugar turned into alcohol, And not just present, it was active, it was multiplying. He watched the yeast cells budding, creating new yeast cells. This was not the behavior of a mere chemical, this was the behavior of life. Pasteur became convinced that yeast was indeed a living organism, a tiny plant-like microbe, a fungus. And it was this living yeast that was responsible for the magic of fermentation. He conducted experiments to prove his revolutionary idea. He showed that if you excluded yeast or killed any existing yeast, then grape juice or beet juice would not ferment to produce alcohol. It might rot or spoil in other ways, but no alcohol would form. He demonstrated that the more yeast grew, the more sugar was consumed and the more alcohol was produced, a direct relationship. This was strong evidence that yeast was not just a byproduct, but the active agent, the living engine driving the fermentation process. This was a monumental shift in understanding, a biological explanation for what had long been considered a purely chemical phenomenon. Pasteur declared that fermentation was the result of life without air. Yeast cells, he explained, could live and grow in the presence of sugar, even without oxygen, and in doing so, they converted sugar into alcohol. This was their way of getting energy in an oxygen-poor environment. This concept of life without air, anaerobic life, was itself profound. It opened up new ways of thinking about the diversity of life on Earth. Pasteur's work didn't stop with identifying yeast as the agent of good alcohol. He also had to explain why fermentations so often went wrong. Why did the beet juice sometimes produce that awful sour liquid instead of alcohol? His microscope had already given him some clues. Those other tiny organisms, the little rods and dots that appeared when things went awry, he suspected these were not innocent bystanders. They were the troublemakers. Just as yeast produced alcohol, perhaps these other microbes produced other unwanted substances, like the acid that soured the vats. He set out to prove this with more careful experiments. He took samples from spoiled vats, rich in these other microorganisms. He introduced tiny amounts of these samples into fresh, healthy beet juice or into a sugar solution that would normally ferment well with yeast. And, the results were clear and predictable. If he introduced the rod-shaped bacteria, the fermentation would produce lactic acid, the same acid found in sour milk and in the spoiled beet alcohol. The liquid would turn sour just as it did in Monsieur Bigot's vats. He discovered that different microorganisms caused different types of diseases in wine and beer. Some produced acetic acid, turning wine into vinegar. Others produced oily substances or unpleasant tastes and smells. Each type of spoilage was linked to a specific type of invading microbe. These were not spontaneous generations, they were contaminants. They came from the air, from the equipment, from unclean conditions. They competed with the yeast, or they took over the fermentation, leading to a spoiled product and economic disaster for the producers. This was a radical idea, that invisible living things could cause such specific changes, such specific diseases in beverages. It directly challenged the dominant chemical theories of the time, and it laid the groundwork for a much larger idea. If tiny organisms could cause diseases in wine and beer, could they also cause diseases in animals and in humans? That question would come later, but the seeds were sown here, in the breweries and distilleries of 19th century France. Having identified the culprits, the tiny microbes that spoiled wine and beet alcohol, Pasteur faced the next crucial question: how to stop them? How could one protect the precious fermentations from these invisible invaders? Or, if they were already present, how could one eliminate them without, of course, ruining the desired product, the alcohol or the wine? He needed a practical solution, one that distillers and winemakers could actually use, a method that was effective and not too complex or expensive. His mind turned to the power of heat. He knew that heat could kill living organisms. It was used for sterilization in some contexts. But boiling wine or beer would drastically change its flavor, its character. That just wasn't an acceptable solution for these delicate beverages, so Pasteur began to experiment with gentler heating. Could he find a temperature high enough to kill the spoilage microbes, but low enough not to damage the taste and quality of the wine or beer? He started by heating samples of already fermented liquids that contained both yeast and unwanted bacteria. He carefully heated small batches of wine to different temperatures for different lengths of time. Then he cooled them and observed. he found that heating wine to about 50 to 60 degrees Celsius, that's about 120 to 140 degrees Fahrenheit, for a few minutes was enough to kill the bacteria that caused souring or other undesirable changes. Importantly, this gentle heating did not significantly alter the wine's taste. The wine remained stable. It did not spoil further even when stored. This process, this gentle heating, became known as pasteurization, a name that honors its inventor and is familiar to us even today. Pasteur first applied it to wine to prevent it from turning into vinegar or becoming diseased. He demonstrated its effectiveness to skeptical wine producers and to scientists. Then he extended the method to beer and later to milk. It was a triumph of applied science, a simple, elegant solution to a problem that had plagued industries for centuries. Millions of liters of wine and beer were saved thanks to Louis Pasteur's insight and his careful experiments. Pasteur's work showed tiny living things caused changes in liquids like wine and beer, which could get sick just like living creatures. Could this apply to animals or even humans? His microscope revealed a world teeming with microbes. If microscopic organisms could spoil beverages, what else could they do? Could they invade a living body and cause deadly diseases? Pasteur's work hinted that microbes came from other microbes, challenging the popular idea of spontaneous generation. This was the seed of the germ theory of disease, a controversial notion that challenged centuries of medical belief. In the south of France, a terrible disaster was unfolding. The vital silk industry was facing ruin. For centuries, France had been famous for its luxurious silks, a source of national pride and economic wealth. But now, the silkworms were dying. The industry was collapsing and thousands faced poverty. This mysterious plague, pébrine, spread rapidly. No one understood its cause or how to stop it. The French government needed help and a brilliant scientific mind. Louis Pasteur, known for his work on fermentation, was urged to take on this challenge. Despite his initial reservations, Pasteur agreed to help. In 1865, he traveled to Alais, equipped with his microscope and determination, stepping into a new scientific battle. Louis Pasteur began his task with diligence. He meticulously observed the sick caterpillars. He saw tiny black spots appeared on their skin, giving the disease its name, pébrine. Many died before spinning silk threads. He examined the tissues of the diseased worms using a microscope. He noticed tiny oval spots, or corpuscles. These were consistently present in the sick worms. Pasteur's investigation was filled with challenges. Through meticulous experiments, he began to see the truth. Healthy worms soon developed pébrine. He discovered pébrine was hereditary. The corpuscles were indeed the culprits, parasitic protozoa. He had finally found the invisible enemy. Discovering the cause of pébrine was vital, but farmers needed a solution to prevent the disease. Pasteur developed a method to ensure healthy silkworm populations. He reasoned that if pébrine was passed from mother moth to eggs, only healthy moths should breed. After a moth laid eggs, she was examined. Infected moths' eggs were destroyed, healthy ones kept. This ensured the next generation was free from pébrine. Pasteur taught farmers to use microscopes. As results improved, farmers embraced Pasteur's method, saving the French silk industry. While Pasteur tackled silkworm diseases and fermentation, the question of spontaneous generation lingered. Could life arise from nothing? Many scientists believed organic matter held a vital force, creating life from non-living substances. Pasteur disagreed, believing microbes came from pre-existing microbes. Life is a germ, he declared. His opponent, Félix Pouchet, claimed microbes grew in sealed flasks of boiled hay. Pasteur suspected airborne contaminants were the source, not a vital force. The French Academy of Sciences offered a prize for definitive proof. Pasteur accepted, confident that rigorous experimentation would reveal the truth. The scientific world watched with anticipation. Louis Pasteur designed a clever apparatus to settle the debate on spontaneous generation. He used swan neck flasks, allowing air in but trapping dust and microbes. After boiling broth in the flasks, it remained sterile. Tilting the flask to allow contact with trapped dust resulted in rapid microbial growth. This proved microbes came from the air, disproving spontaneous generation. Louis Pasteur's swan neck flask experiments delivered a fatal blow to the doctrine of spontaneous generation. Life, it was now understood, only came from pre-existing life. omne vivum ex vivo. This shift would have far-reaching and transformative consequences, especially for the practice of medicine. Pasteur laid a solid foundation for the modern germ theory of disease. It became clear that many diseases were caused by specific invading microorganisms or germs. This was a truly revolutionary concept. This would lead to critical advancements like antiseptic surgery and a greater emphasis on better hygiene. These measures would go on to save countless millions of lives. He had clearly shown the world that an entire invisible universe of microbes played a critically important role in both health and disease, thanks to the genius of Louis Pasteur. In the 19th century, rabies struck terror. It meant certain agonizing death. Transmitted by infected animals, mainly dogs, once symptoms appeared, there was no hope. The disease led to madness and paralysis. Even the sight of water could trigger convulsions. Countless remedies were tried to no effect. Louis Pasteur, having conquered diseases in wine and beer, turned his mind to rabies. Could his methods unravel this dreaded killer? Pasteur was driven to alleviate suffering. Louis Pasteur confronted rabies head-on, understanding the suffering it caused. His colleagues warned of the risks, but Pasteur was resolute. His laboratory became a battlefield against this deadly foe. The challenge, the infectious agent, was too small to be seen. He examined saliva and blood, unable to identify the microbe. Today we know it's a virus, but this concept wasn't understood then. He experimented, often with rabid dogs, facing constant risk. A single bite could mean death. Pasteur and his team's courage was extraordinary. Driven by belief in science and compassion, he relentlessly pursued a solution. Pasteur observed rabies' devastating effects on the nervous system, pinpointing the brain and spinal cord as primary infection sites. He hypothesized the infectious agent multiplied within these tissues, guiding his research. Using brain tissue from deceased animals, he induced rabies in rabbits, creating a consistent disease model. A breakthrough came by attenuating the virus. Drying infected rabbit spinal cords weakened the agent over time. This created a graded series from highly virulent to almost harmless, an arsenal for developing a protective vaccine. Louis Pasteur tested his hypothesis. Could weakened rabies protect against the deadly disease? He experimented on dogs, often rabies carriers, aiming to break the transmission chain to humans. Dogs received injections of weakened rabies, then were exposed to the virus. Unvaccinated dogs died, but vaccinated dogs remained healthy, protected against the madness. Pasteur's method worked, a major breakthrough. Could it be used on humans? The leap was enormous. Fraught with ethical concerns, the world waited. In 1885, Pasteur's rabies vaccination of dogs offered hope, but he hesitated to use it on humans. On July 6th, a desperate scene unfolded. Marie-Angélique Meister arrived with her 9-year-old son, Joseph, who had been savagely attacked by a rabid dog. The local doctor offered no hope. Joseph would die. She pleaded with Pasteur to save him. Pasteur, in turmoil, examined Joseph's wounds. His colleagues urged him to try his treatment. After a sleepless night, Pasteur made his courageous decision. He would try to save Joseph Meister. The treatment of young Joseph Meister began on the evening of July 6, 1885. Pasteur, assisted by Dr. Grancher, commenced the procedure, injecting Joseph with an attenuated rabies agent. Over 10 days, Joseph received 13 injections, each with a more virulent strain. The following days were filled with unbearable tension for Pasteur, constantly worried about Joseph. Weeks passed, and Joseph showed no signs of rabies. The treatment had worked. News of Joseph Meister's survival spread, and Louis Pasteur was hailed as a hero. Humanity now had a weapon against this dreadful disease, a beacon of hope for all humankind. Following Joseph Meister's recovery, the news spread quickly. People arrived at Pasteur's laboratory, desperate for treatment. Another case was Jean-Baptiste Goupil, who fought off a rabid dog. He too was successfully vaccinated, solidifying public confidence. The demand became overwhelming, requiring a dedicated institution. In 1888, the Pasteur Institute was inaugurated in Paris. The institute became a world-renowned center for microbiology. Pasteur's rabies vaccine transformed him into a global icon. Countless lives were saved thanks to his courage. His legacy revolutionized medicine and public health forever. Louis Pasteur's journey, though brilliant, faced skepticism and resistance. Established scientists and doctors were slow to accept his findings on fermentation and germ theory. His calls for hygiene were dismissed. Pasteur trusted his experiments, inviting critics to witness undeniable demonstrations. He was a courageous advocate for a new understanding. Louis Pasteur faced a fierce battle over spontaneous generation, the belief that life could arise from non-living matter. This idea undermined Pasteur's germ theory, suggesting microbes could appear without a parent organism. His opponent, Félix Pouchet, believed in spontaneous generation, but Pasteur suspected contamination in Pouchet's experiments. The French Academy of Sciences offered a prize to settle the debate. Pasteur devised the swan neck flask experiment. The flask allowed air in, but trapped microbes, keeping broth sterile and less exposed to dust. This proved life comes from life, a major clarification in biology. Louis Pasteur's dedication demanded immense personal sacrifice. It took a significant toll on his own health and personal life. In 1868, Pasteur suffered a severe stroke, leaving him partially paralyzed, but his scientific spirit was unbroken. Pasteur endured profound personal tragedies, losing 3 daughters to illness. These losses strengthened his resolve to fight the diseases that brought sorrow to his family. He transformed personal anguish into a powerful force for scientific good. Pasteur faced a harrowing choice: risk an untested rabies vaccine on young Joseph Meister, or let the boy die. The world watched, critics circled, and Pasteur, a chemist not a doctor, stood at the center of a storm. Against fierce skepticism and immense pressure, Pasteur pressed on, driven by compassion and conviction, supported by trusted colleagues. His courage in this crucible would change medicine forever. Even after saving Meister, Pasteur's work was relentlessly challenged. Critics seized on every doubt, forcing him to defend his discoveries in public and through dramatic experiments. But undeniable results, like the anthrax vaccine trial, silenced skeptics. Pasteur's perseverance transformed medicine, saved lives, and proved that fighting for scientific truth can change the world. Pasteur's death marked not an end, but the beginning of a global movement. The Pasteur Institute and its offshoots became beacons of research, spreading his rigorous methods and humanitarian vision worldwide. His story inspired generations, turning science into a heroic pursuit and his principles into the gold standard for research, industry, and the fight against disease. Pasteur's germ theory revolutionized medicine, transforming surgery, hygiene, and public health. Inspired by his work, Lister's antiseptics slashed surgical deaths, and new hygiene standards saved countless lives. Pasteur's vaccines laid the foundation for immunology, proving that humanity could fight and conquer its deadliest foes. His legacy is woven into every act of modern preventative medicine. Pasteurization protects our food, and his insights into microbes power industries from antibiotics to biofuels. His scientific rigor set the standard for research across all fields. Even his early work on molecular asymmetry shapes modern medicine. Pasteur's curiosity unlocks secrets that still drive innovation and safeguard our health today. In a world facing new pandemics, Pasteur's principles, rigor, perseverance, and faith in science are more vital than ever. His journey from crystals to vaccines is a timeless lesson in the power of curiosity and resilience. Pasteur's legacy lives on in every vaccine, every sterile instrument, every safe meal. His light guides us still, inspiring hope and progress for generations to come.